Hello, everyone, and welcome to today's live broadcast, Rebuilding Evolutionary Success using Inscripta's Onyx Digital Genome Engineering Platform to ease and enhance adaptive laboratory evolution experiments. I'm Alexis Krause of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Inscripta. For more information about our sponsor, please visit their website at inscripta.com. Now, let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. If you have any trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. I'd like to now introduce our presenter, Dr. Tyson Shepherd, Scientist 2, Applications Development, Inscripta. For a complete biography on our presenter, please visit the Biography tab at the top of your screen. Dr. Shepherd, you may now begin your presentations.
Hi, thank you for that introduction. So like was just said, I'm Tyson Shepherd. I'm an application scientist here at Inscripta. I began at Inscripta about approximately 6 months ago, and what really drew me here to Inscripta and to the platform is to see these capabilities and really the desire or the wish that I had these same capabilities when I was in graduate school or when I was doing my postdoc. I see a lot of potential for these impacting. It's a little like, as you'll see, it's a little like there's a thesis in every run of the experiment, and we're really bringing this to all the laboratories. So my presentation, I'll be going over both the platform at the start, but then also additionally, we'll be showing an application of this in adaptive laboratory evolution and how we've recapitulated these libraries. So to begin with, for the overview of the platform, the fundamental basis of the platform is founded on the concept that of co-delivery of a guide RNA, a CRISPR, and a donor template all in one that then can simultaneously be barcode tracked inside the cell. And that barcodes then allows you to then segment out the populations and later do all kinds of interesting discoveries. And really, built on that fundamental basis is the concept that we want to democratize biological engineering to really bring this biological engineering to every lab that we can. And to do so, we're not just helping design these barcoded editing plasmids, we're simultaneously also designing elegant and efficient hardware and software that eases the overall process. A single box that will provide, you get the reagents and then it does the CRISPR editing, and then you retrieve the cells for your subsequent experiments. And then our technology is also based on the CRISPR enzyme MAD7, which is similar to CPF1 in a way, but is quite different and has some unique features and is also capable of sort of freedom of use. The sequence is available and labs can use this readily. So historically, the way that mutations have kind of been played out in molecular biology is that you can kind of split this apart into two different axes, which is you can edit a small number of genomic locations, for example, do gene knock-ins or knockouts, and then you can do subsequent protein engineering at certain locations. You can also, maybe on a different axis, you can think of the variety of different edit types that can be made. So you can do whole pathway engineering, you can do this protein engineering, you can do site-directed mutagenesis. but those are all low in the number of locations that can be made. Simultaneously, you can have relatively low number types of edits that can be made, but across larger genomic landscapes. What would be fascinating is imagine the capability of being able to do genome-wide engineering rather than individual pathway or protein engineering. And then you can explore the entire breadth of what's allowable by the genome just by having the capabilities of editing. And historically, this has just been compressed away from capable just because of the inability to track so many different modifications. Well, here, because of the software that's being made and the automated aspect of the instruments combined with the power of barcode tracking, we can actually explore the full potential of genome engineering. So not just in one locus anymore, not just of one type of modification that can be done, but really anything that you can imagine can be spread out across the genome. So to do so, we kind of stick to that engineering concept, which is the design, generate, test, learn cycle. So really the fundamental begins with that design. We have this easy to use web portal to be able to make your modifications and interact with the genome in ways that you've never had the capability to do before. This allows you to then engineer in modifications into your cells that then after once you order the reagents, after a few weeks, a kit arrives that you then load into your instrument. The instrument runs, and then you're able to get out like a single tube that contains your edited cell population. Then you can take that tube and go do your experiments. You can do your selection experiments and do all sorts of downstream phenotypic-based experiments, and then come back and you can use sequencing to be able to track those barcodes or the edits on the genome. And then you can pull that back into our software that we will then help analyze for you and provide the results from these editing experiments, so that then we can help you parse through all of this data that you'll be getting off of the experiment. So at the start, we'll be offering both editing in E. coli and Saccharomyces cerevisiae yeast strain, and then we have some Inscripta standard strains, both E. coli and yeast, that we can work with quite readily. And then we're also having some capabilities of bringing on custom strains that are from clients, that are provided from clients, and being able to have the same operational infrastructure come in onto your own strain of interest. So for right now, though, we're starting with E. coli and yeast, and right now we're also at launch, we'll have one edit per cell, but we'll be expanding in the future. The edit types that can be done are insertions, deletions, and substitutions. And per library, per run, you can have from 100 to 10,000 different edits across your genome if you're using E. coli, and from 100 to 6,000 in yeast. So large landscape to play with. Per run, per sample, we will typically see 30% edited cells. That's complete and intended edits on those cells. And that's somewhat variable just based on the type of edit that you're doing, just how large the insertion is, for example, and so forth can have an impact on that. From the time to order to receive the reagents is about 3 to 4 weeks. And then the instrument time that it's running is only approximately 1 to 3 days before you get this output vial that contains all of your edited cells. So as I mentioned, there's a wide diversity of possible editing types, both in coding and non-coding regions. So those, just to get an idea to have in your head about the different editing types that can be made. In non-coding regions, for example, you can do up to 61 base pair insertions, and those include a wide series of promoters you might think about as being one area of interest. So you can do an insertion outside the coding region. You can certainly do insertions within coding regions. You can also do substitutions. So here I show an example of a 38 base pair substitution of terminators, and that then is, of course, swapping out or replacing the nucleotides with those that you've programmed in. Additionally, in coding regions, now I just highlight all of those things are still capable in coding regions, but we also, have proven out the capabilities of generating knockout strains by triple stop codon insertions into or across the genome. And we can also do saturation mutagenesis across coding regions, so you can really explore the breadth of protein of interest that you're studying to see how the cell responds to it, or to see variants of a particular protein within cells. So one way to think about this, or a great way to kind of conceptualize what we're doing, is that we're taking the old paradigm of having to make a 96-well plate with individual edits by introducing the guide RNA and the donor template into each of those individual wells that aren't combined together or linked together. And so they've, by necessity, had to have been separated into these wells. And with our technology now, the way to think about it is that we've taken that 96 partitions, those 96 different wells, we've condensed all of that biochemical information that you can get out of that down into the single cell, so that every single cell becomes its own little reaction space. We're still delivering a single guide RNA with the donor repair, but then additionally with this barcode allows us to track the individual cell populations, which then allows it to be very similar to this 96 well partitioned format. So we can do, if you remember, we can do 10,000 different custom designs on a single run on any across a population of the E. coli, for example. What this leads to is millions of precisely engineered single cells that you can then take downstream and generate the phenotypic response from all of these mutations that you've made into the individual cells. So now I want to highlight an application of this particular technology, and that's to adaptive laboratory evolution. So we've been working with Marcus Ergaard's, a leader in the field of adaptive laboratory evolution, to obtain a set of mutations that are found through AL experiments, and then recapitulate these using the Onyx technology. And I'm going to present that data here. So if you're not familiar, adaptive laboratory evolution, or AL experiments, is a high throughput way of being able to generate mutations in strains due to, or when you have them assaulted with some chemical that would otherwise inhibit their growth. So it's a very powerful approach that under continuous growth in these stress environments, you can get these mutations. And then by the powers of next generation sequencing, it really enables you to then go into that population and identify the mutations, the single nucleotide polymorphisms or insertions or deletions that are then discovered and that can then have applications both in biotechnology for the evolution of strains that function under a given chemical stress, but then also can give rise to new biochemical information. However, the ale, it has a couple of drawbacks to it in that it's time consuming. It can take weeks to run an experiment, up to kind of tongue-in-cheek, it can run for decades to be able to get explore the mutation space. It does require typically rebuilding that strain for downstream characterization for new biochemical understanding, as well as having that engineered custom strain. Then it also, ale is fairly restrictive in its evolutionary flow because it's moving linearly along a path of evolution. So it doesn't get to explore the full breadth of being able to try multiple different modifications because it's just moving linearly along what wins, kind of a random walk in a way. The Inscripta Onyx platform actually enables a couple of interesting features that can add on to these ale experiments. So it's a high throughput method ultimately of generating a massive number of variants that can then act as a precisely engineered initial population. So you can come at this with the knowledge of the mutations that you do want to study, and that's sort of what's shown here in this work. But then you can simultaneously use this to ask a lot of new questions, because then you can have your starter population that you know precisely what you're starting your ale library with, and then you can make these modifications and then grow out from there so that you're no longer restricted to that strict evolutionary linear path. So we really think that this provides a lot of power to the ale workflow. So for the overall workflow of the Inscripta Onyx platform is that then you work with the interface, the user interface, to design and generate new modifications that you want made to the genome. In 3 to 4 weeks, you get your reagents plus an additional day of editing, and then you have a tube of engineered cells that you can then take for downstream selections. You then process those for sequencing. You can upload those sequences then for analysis of your barcode selections that you've done, and learn new things about your experiments of interest that then can feed back, and you can actually then go through this cycle multiple times and generate a new powerful strain for the chemical of interest, for example. So in this case, we did a high throughput rebuilding of ale variants. So again, working with Marcus Hergard and his lab, we generated a list of 372 different ale variants that were previously found through by growth within 11 different chemicals. Of those 11 different chemicals, there were 2 chemicals to note here, which is p-coumaric acid and isobutyric acid. Those then ale variants were taken forward and used on our design platform to be able to rebuild those variants on our using the Onyx technology. Further, because of the wide space of what's available for modifications, we might also be interested in what would happen if we inserted mobile elements or made deletions or knockouts within the genome of those same genes or others that have been shown to be modified. So of these 372 variants that were done, those are across all 11 chemicals, and not just p-coumaric acid and isobutyric acid, but this is across all the different chemicals. So we can really test widely the biochemical information from each of those. This brought us to a total of 992 individually designed and trackable edits that are going to be made to the genome. So this is now an actual picture of what you might see when you're using the Inscripta platform to edit your genomes. So it's a very intuitive GUI interface here where you can interact with the genome. I almost think about it as it's a little bit like if you were able to SSH or telnet into the cell and be able to, interact with it or program it. It's sort of that nice to work with. Then you can make your insertions, deletions, and substitutions, and then they show up both in the E. coli coordinate system, for example, on the left, as well as we have a genome viewer that allows you to then zoom in and be able to see the edits that you are planning to make to both ensure that those are the edits that you want, as well as be able to, in real time, get feedback for which edits would be most likely to be functional within the cells. So then, as I said, downstream from that is once you get your tube of cells, then you take that on to sequencing. And because of the sort of the magic of the barcode, that allows you through sequencing to be able to investigate which variant is actually present within your pool then. So each of the cells are going to contain a different guide RNA donor and barcode. By the analysis of the barcodes, we can really map those back onto the E. coli coordinate system. And on the edits that are made, this is kind of then, flashing back to the GUI, it aligns perfectly well with the edits that were made. And it turns out that we see within our sample, when grown in LB, 92% are observed. And some of those that drop out, some of those unobserved, are just potentially due to partially to cell viability. So some of the knockouts that we make are going to be within the transcription machinery that just might simply not be compatible. And you might see the same thing, and that alone is interesting biochemical information. But we can then take this fully functional library with 92% of the edits that we've made and carry that forward and be able to continue to track those individual barcodes under different chemical assaults. So let's do that now. So we take this on that engineered population, we take that on to the next step, which is our selection experiment. So in this case, we provide then a selection and then goes on to sequencing. So we take that single tube of library that contains all of those edits within it, the 992 different edits, and we set up the experiment as such. We have our control, our base control, which is the M9 glucose. That's with no addition of chemicals. Then using, again, Marcus Ergaard and his lab's work, using concentrations of p-coumaric acid and isobutyric acid of 7.5 grams per liter, which is enough to start to have a growth defect. We also put the same, inoculate from that same pool into the p-coumaric acid and isobutyric acid. Additionally, to be able to test whether or not it's a salt effect of any of the growth we see, we also put it into NaCl, 400 millimolar, and we spike it into LB to see under conditions where there's no restrictive growth like there would be under M9. So now when we have this selection experiment and we then do barcode analysis and get the sequencing from that information, that then feeds on to the next step, which is really where the true joy of science comes, right? Which is in the analysis and learning that we get from the data, and then the new biochemical knowledge that we can gain from such experiments. So that analysis population, the first thing that we did was to assess the quality and reproducibility of the data. We then identify, looking at differences between isobutyric acid versus p-coumaric acid versus the M9 media, to be able to investigate what are the mutations that make the most difference within each of the growth populations. We look for these trends of enrichment and depletion that then give us biochemical information about the individual edits that have been made and their performance within that particular chemical. And then we functionally assess these enriched and depleted genes, and that's where we gain our new biochemical insights and add to the existing knowledge of these individual strains. So this is one of the graphs that's common with this data, it's a plot of the log of the relative abundance of a barcode in a particular sample after growth versus in a chemical versus in the control sample. So the relative abundance is how much of the individual barcode is in the entire pool of barcodes. And so each of these 4 plots are the 4 different chemicals that we tested against along the x-axis is the log 10 relative abundance in M9. So you can see an interesting effect for NaCl. You see that it's quite condensed along a line. This indicates there's not much, there's a significant amount of correlation of each individual edit's growth within both NaCl versus M9 alone. In LB, it gets dispersed a little bit, and you can really start to see additional dispersion with the coumaric acids and isobutyric acids. And so we're starting to see like the populations fall away or become enriched in these. And that's where these edits become can become quite interesting as we look at this. So this gives us an overall snapshot of the correlation and outliers, and those outliers are the ones that then we would want to look at for effects within a particular chemical. So each of these experiments, as I mentioned, was done in biological triplicates, with the exception of N9, which was done in replications, 6 replications. Along the diagonal, you see the extreme correlation between any given experiment, and within the triplicate measurement, it's highly correlated. And you can see examples then of, like, for example, M9's behavior in NaCl. We saw that it fell well within a line. And here, this is now the Spearman correlation matrix that shows this correlation as a measure of what you're seeing. And really, you see that high correlation coming through with that NaCl to M9 conditions. So all the strains are behaving the same way, whether or not, irrespective of the presence of NaCl in this instance. You see the dispersion start to take place within isobutyric acid and coumaric acid. So you can kind of see that escape away from the M9. But then when you look at coumaric acid and isobutyric acid responses individually, they're not very well correlated at all to the growth response for every one of those individual cells that were edited. So if we just look at that relative abundance plot, that log relative abundance plot, again, we can see that dispersion as well, that there's a number of outliers between these 2 different conditions. One additional way of looking at this same kind of separation of data is by doing a principal component analysis. So this is the PCA analysis of just the 372 AL edits. They, as you can see, they cluster on the replicates. So these are all the replicates plotted out here, and they cluster in distinct locations within the PCA analysis. So there's a clear separation of each of the individual chemical stresses that have been placed on this, so the population is behaving differently. And with the replicates, you can have a high confidence moving forward that you're getting reproducible data. So not only are they behaving distinctly, but they're reproducibly, very nicely reproducibly behaving in a distinct manner. And so then this is now the, these are really like looking at the entire population of all of the cells within the individual stress that was placed on them. So then what we can also do is look at the individual edit that was made and be able to see any biochemical patterns that might show up. So this is an example. This is on then the E. coli coordinate system, maps all of the edits. And so each of these dots on this radial plot are representative of one edit that was made. And this is now an average of the triplicates that were done. And along the radial direction outwards is a measurement of the enrichment of the edit or depletion. So the 0 line is in blue, is surrounding this radial plot. And then the distance away from that is representative of the treatment abundance, which you saw, divided by the control abundance, and then it's the log of that. And that gives us an enrichment score of that treated condition. So this is for coumaric acid. You can see that there are certain mutations within the cell population that give a rise or an enrichment within the coumaric acid. And there's a number of also mutations that are depleted out in coumaric acid response. So they become more susceptible than M9, which is the control. They become more susceptible with these mutations being made. Then, as plotted here, you can kind of see that the reproducibility of this data is so pristine that there's fairly few points that show up, but this is a p-value of those that are colored are a p-value of less than 0.001. So it's quite reproducible, and the point size here then indicates the actual number of counts. So you can have an edit which might have low counts but be significantly enriched, but simultaneously it's possibly even more exciting when you see multiple counts showing up and also significantly enriched. So this then is looking at isobutyric acid response rather than the coumaric acid response, or side by side. And you can actually already start to see that there's significant differences in responses between the two different chemical stresses that are observed here. So for example, I might point your eye to like the 4 million base pair region, which is kind of maybe close to the origin of replication. You can see within there's some edits that are quite different, where it's significantly enriched in isobutyric acid, and some that are enriched and some are depleted in the coumaric acid response. So how do you unpack this data? With all of this data, there's really great plots that can be made with this. Another common plot that you might have seen if you've come from the RNA sequencing world is like a volcano plot, it is sometimes called. So in this plot, it's the effect of enrichment or depletion is essentially made along the x-axis. So that's the log of the abundance of the coumaric acids, barcodes over the M9 response. And so on the left is coumaric acid, on the right in green is isobutyric acid. And then on the y-axis is the log of the p-value. And so you can have higher confidence than in those that are high on the y-axis. So really what we see on the, let's focus on the coumaric acid for a moment, is I've highlighted here a number of individual mutations that were made in the annotated genes here as being significantly more resistant to coumaric acid pressure when these modifications were made. As I mentioned, each of these points is an individual mutation. So you can see, for example, that rpoA is showing up in multiple different locations. So those are different mutations within the edited cell population. And we can just track those. And so then you see with isobutyric acid, the same type of thing where you can have rpoC or rpoB that are showing up that can have a substantial effect on the growth, or essentially is enriched within isobutyric acid. But you see that rpoA is not showing up as one of those edited populations that's highly enriched. So let's focus in on this. The rpoA is part of that RNA polymerase machinery, and so here I've actually drawn out even more to make it clear is some of the actual mutations in rpoA that coumaric acid response gave us. And so these individual point mutations that are made are, we can do insertions, which I've shown that are recapitulated from an AL experiment, individual point mutations, and then also those individual point mutations can be either enriching or depleting. And we see that in this case is that rpoA has some mutations that are enriched and others that are depleted in the coumaric acids. And so what we're seeing here is that then also on the right, I might point out is then the isobutyric acid, which has that rpoC, which is also part of that same complex, the RNA polymerase machinery. That rpoC is showing up as highly enriched in isobutyric acid, but rpoA and some modifications of the rpoB are actually then depleted within the isobutyric acid response. So this is sort of one of the powerful things is that so far we've been looking at population-wide responses when put under chemical strain. But this gives us something new to think about, which is how can I use this technology to look at individual proteins and investigate responses for mutations and substitutions that are made within an individual protein. So what we did here is we actually mapped all of the modifications of the rpoA from the coumaric acid response onto the PDB structure. So this shows the three-dimensional structure on the right of the RNA polymerase, and highlighted in wheat color on the right is the rpoA subunit of the RNA polymerase. And on the left, we show a volcano plot again, but now this is the all individual point mutations within the rpoA machinery. So in color, in green are those mutations that were made that made the cell that those modifications are made in more tolerant to coumaric acid. And on the left of that plot in red are those mutations that were made that made it less tolerant. And just to help clarify better is that in gray are dots where was the isobutyric response. So in almost all cases, as you can see in gray, is that the isobutyric response was depleting with all modifications. But in coumaric acid, some were improved tolerance and some were depleting. Mapping those on now to the right structure with that rpoA shown in wheat, we can see how those mutations would impact the amino acids, and that gets colored now onto the RNA polymerase. And so we see that the mutations that make the cell more tolerant to coumaric acid are colored in green and clustered into a specific region on the N-terminal domain of the RNA polymerase. Meanwhile, those mutations that make it less tolerant to coumaric acid are clustered on the C-terminal domain. What's known about the RNA polymerase is that N-terminal domain is typically associated with subunit assembly and dimerization of the rpoA, and then the rest of the subunit is coming together. So it's kind of a structural protein in a way. Meanwhile, that C-terminal domain is responsible for interacting with translation factors and giving that stress response. So one can imagine that the negative tolerance that's associated with mutations on the C-terminal domain might be because it's disrupting its capability of having that stress tolerance from interacting with these transcription factors. Yet the more tolerant mutations might make it easier to assemble, or it might very well be a binding site, for example, of coumaric acid. So that's where more biochemical characterization might come into play. But it does give you a sense of the power of what can be done when you're doing these type of assays to be able to do genome-wide screening for modifications and being able to dig down into very specific biochemical questions to then achieve new hypotheses that you can carry forward, maybe for drug screening, maybe for structural biology and being able to identify new variants that you might be interested in testing, or for just simply better understanding the evolutionary pathway that you typically see in your own AL experiments and see that this is now playing out in a way that we can get a snapshot across many different modifications simultaneously. So I also want to give you a sense that, we've mostly been talking about just those Ale-specific modifications that were discovered through the Ale experiments, but we also have this rich data set of frame shifts that were made by mobile element insertions, as well as deletions that we can kind of test loss of function for those Ale mutations that are made, but also simultaneously get a whole new deep understanding of when those proteins are knocked out, what's their own behavior within the same kind of chemical stress. And you can see that some of those modifications are more resistant for deletion. When you put deletions and frame shifts in, you can kind of see that those data sets alone are rich and provide a wellspring of their own biochemical information that comes from those. Generally, what you can do then is kind of looking at that whole population of deletions and frame shifts plus the Ale edits, is that you can sort those. This is just broadly sorting those out into different functions. So metabolism, membrane response, regulation. Is it part of the replication machinery, the transcription machinery, the translation machinery, or is it transport, or is there other? And really, one more thing that we just don't have time to talk about are all the unknown genes that come up in these Ale experiments, where this can give new insights into the function of unknown of genes with unknown function. So the so-called Y-ome, for example. So here plotted on the y-axis is that the abundance, again, the log of the abundance, so that enrichment score. And then along the x-axis are these categories of different. And so then each of these strips are individual genes within the study. You can kind of see global trends. So for example, modifications in the membrane or in the replication or also in the other category are generally depleting for the p-coumaric acid response. But in transcription, as we've already seen within the rpoA story, is transcription can have a substantial effect for enriching modifications within the transcription machinery. So we really see that capitulated here, and this can be done for multiple different stresses then, like for isobutyric acid, and we can see the effects. And that gives you a sense of what are the impacts of globally of the different machinery within the cell. Further, this is also quite helpful in that it's these modifications, if you remember, were recapitulated from an original library that were generated from the Ale experiments. And here we see some of these same modifications showing up in our own response from our Onyx edits. So every other column, there's the isobutyric acid from the Ale study. This is the most common, is PYKF modification, and the next most common is the rpoB edit that's being made. So these show up again and again in Ale experiments under isobutyric stress. We actually then see rpoB, an individual point mutation, can have a substantial effect on the enrichments of the isobutyric acid. So in this dark color is eightfold or greater enrichment in isobutyric acid compared to that M9 control. And again, in lighter blue here now is a fourfold enrichment, greater than fourfold enrichment. And so we see that response with a couple of different mutations within rpoC along the isobutyric Onyx column that show up, that's also seen within that Ale library that a couple of times rpoC modifications show up. With p-coumaric acid, again, mutations within Rho show up repeatedly in 7 out of the 8 replicates in the Ale experiments, whereas in the Onyx platform, we see that mutation within Rho. We see a specific mutation that provides the most tolerance in p-coumaric acid. So again, the dark color is indicative of a greater than 8-fold enrichment over M9 control. We also see these other genes of interest showing up, rpoA, and you can see that there are a few examples of genes of unknown function that do show up as well. That gives us a better biochemical insight as well. So we kind of see that recapitulation of those AL results already, but we also see the diversity that this engineered population actually allows us to investigate single point mutations simultaneously with being able to broadcast a wide net of modifications so that we can really capture an entire landscape for a stress response for a given chemical. So the overall workflow here that we talked about is we take that edited cell, we either can take it to isolated cells and look at responses, or take whole pooled samples, which is what we did in this case. And then we can downstream of that, we can screen for either phenotypic responses with direct detection of behaviors of the cells or proteins. And then we can also take all of those to sequencing and do a genotype on it. And so we're really finding ways to wrap together the phenotypic response with the genotype, the underlying genotype. And that allows us, to open the doors to all these new discoveries. So some of those top hits for that diverse library that was generated, you can imagine then it can be brought forward multiple rounds, so that if you're really interested in getting the best possible strain to behave under a compound, you can carry this same workflow through and feed back those modifications. And this really stretches its legs when we introduce our combinatorial editing workflow, which will allow multiple different edits to be able to be made within a genome simultaneously. And then you can build upon those modifications that you've seen previously and see increases in stress tolerance to fantastic levels as you make these modifications and really broadcast mutations across the entire genome. So we've really tried to, in this study and within the actual Onyx platform, we've shown that, we've made it so that you can make thousands of modifications within an edited pool of cells that we're trying to make easy for you to be able to just do a push button kind of service of being able to broadcast these mutations across the genome. In this particular study, we showed that we can recapitulate AL study using modifications that were found within previous AL experiments, but then also really this showcases the ability to then take that engineered population that precisely what's in there and carry that forward for subsequent AL experiments or subsequent Onyx editing experiments to be able to combinatorial edit in the future. And then finally, this took just a few weeks at the bench when we were able to recapitulate this study that was took several months to be able to carry out under stress tolerance. So we've really eased the overall workflow as well with our single edited population as well. So I think this really addresses all of the needs of the AL studies, and I think it could really advance this entire field, as well as you saw, the impacts on structural biology and screening. So thank you very much for listening, and I would be happy to take any questions now.
Thank you, Dr. Shepherd, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Our first question is going to be a two-part question, Dr. Tyson. How applicable is your technology to the genome editing in non-standard microbes? And is this platform compatible with genome engineering in eukaryotes such as yeast?
Hi, thanks a lot for the questions coming in. So this is a great question. Thanks for asking it. So at the time of when the instrument is being rolled out, we are principally supporting E. coli, both internal strains as well as capabilities to explore alternative strains. But then also we're supporting yeast in particular. So S288C strain is sort of the standard strain that we're working with. But then also we're interested moving forward with expanding and testing other E. coli and yeast strains and expanding over time.
Great. Now our next question is, are you engineering the population first, or are you creating a true mutant population through ale that can be analyzed on the back end?
Okay, thanks. That's another great question. This presentation was kind of highlighting both of those applications. However, to clarify, is that the library that was that the data comes from here was generated originally from an ale data set. So they were originally, all these mutations were raised in an ale experiment against multiple different chemicals. But then we took those sets of edits and then engineered them directly into the population. So the population and the data that you're seeing here is actually composed of an engineered population, and then subjecting that population to these two specific chemicals.
Now, how do you control cross-contamination during your high-throughput analysis?
So this comes from, you can use amenable technologies to next-generation sequencing to ensure that there's not cross-contamination within the actual sequencing to the limits or capabilities of the sequencer that you're using. In this case, since our main or our major readout in this particular study was barcode amplicon. So that means that there's a barcode that's present not on the genome, but separate from the genome, that's of a length scale that's easily read out with Illumina-based sequencing. And then from cross-contamination, we don't have to worry so much from different cultures, but then those are each prepped individually and then sent for sequencing with different indices. Yeah, thank you. That's a great question.
We do have time for some more questions, but I do want to remind our audience that those questions we are unable to answer today, and those that come in during the on-demand period, will be answered by Dr. Shepherd via the email address that you provided at the time of registration. So let's continue on to our next question. What is the best use of your technology? What is the best use of your technology would be for a higher protein or enzyme production?
That's a nice question. Thanks for asking that. That's an application that I think is particularly interesting for this technology, because you've seen even with the data shown here that you can make variants within a protein itself. You could imagine that you might be able to apply this technology both for codon optimization of a particular gene that's integrated into a genome. You can modify promoters. We've done that in other apps that have been spoken about that you might enjoy going back and reviewing. So you can modify promoters, ribosomal binding sites, and mRNA, but then also simultaneously you can make genome-wide modifications that can possibly boost the potential of protein production by changing the capabilities of the metabolic rates or amino acid production, or even the translation machinery itself can be modified to allow for increased expression. Another thing that's sort of interest with enzyme production is that you might see some improved effects just by increasing or allowing for production of resistance to the chemical that's being produced. So all of these are possible, and it's a great application to explore. Thank you.
Let's go to our next question. It looks like this one is a two-part question as well. I'm having a little difficulty understanding the generation of libraries. Does Onyx generate the barcodes that makes this so powerful for constructing genome-scale libraries? And does it synthesize the DNA itself and transform it, or is it really just a design platform?
That's an excellent question. Thanks for the opportunity to clarify on this. So the barcodes are not something that you'll directly interact with. That's something that's automated and through the design platform itself. Those barcodes are just used for the actual readouts then. The actual instrumentation is not synthesizing the DNA. What you'll receive when you order a library is a set of components that you'll plug into the instrument, and the instrument takes care of all of the cell handling itself, such that you put in these components and kind of press the start button. You know, I'm kind of overgeneralizing here, but it then automates the entire process and workflow for growing out those, making those edits, growing out, and then you'll receive a test tube back with a library of those edits in the actual cell culture. But the barcodes themselves are distinct and separate from the from on the genome. They're off the genome and can be tracked for subsequent sequencing. Thank you.
Now, Dr. Tyson, our next question is, can you remind me how you made the IS mobile element insertion and how long of the edit can be made?
Yeah, good question. So the length of the edit that can be inserted is up to 61 base pairs. And we have, as I mentioned a minute ago, we have a promoter library that's the subject of a former presentation you can see by Dan Held, for example, who's talked about such an application. So insertion lengths can be up to 61. In this case, the mobile element here, we're really just taking it kind of as a synthetic mobile element. So this was 35 bases, so that would induce a frame shift. But it's similar to sites where mobile elements were introduced within the AL studies, which is a common motif that occurs. And so we wanted to take advantage of that and still use the breadth of our platform. So here we were just using synthetic, maybe put quotes around it, mobile elements to get that breadth of edits. Thank you.
And it looks like we have time for one more question. So earlier you mentioned that an approach with Onyx can be different than AL. How can this approach take a different path than a linear introduction of edits?
Oh, that's an awesome question. Thank you. So the way that occurs is that because you're starting with an engineered population, so going through the workflow mentally is that you've went onto the portal, you've made your designs and interacted with the genome and come up with a set of designs that you're interested in, be it of a particular protein or set of proteins or pathway that you're interested in, but maybe across the whole genome. You can then get the tube off the Onyx instrument that has that edited pool population, and by sequencing and the way the technology works is that now you have an engineered starting population that you still know exactly what's in it. Just like when you're starting your experiment from a clonal isolate, that's what you really care about, is that you know exactly what you're starting from. Here you still know exactly what you're starting from. It's just now you can then go on to do an AL experiment with up to 10,000 different edits across the genome. So you can really get like a full breadth of not just those single edits like what was shown here, but then you can do that simultaneous AL experiment and raise new mutations against the edited population that you can go back and track. Thanks. That's a great question. Thank you.
Thank you again, Dr. Shepherd. Do you have any final comments for our audience?
No, thanks a lot for listening, and please email me or let me know if you have any questions about the technology. And then, as mentioned, we'll hopefully be in touch if you've asked a question that I haven't had time to get to. So thank you very much.
Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Like Dr. Shepherd said, questions that we did not have time for today and those submitted during the on-demand period will be addressed by Dr. Shepherd via the contact information that you provided at the time of registration. As a reminder, there will be a survey at the end of the presentation, and we look forward to your feedback. We would like to once again thank Dr. Shepherd for his time today and his important research. We would also like to thank LabRoots and our sponsor, Inscripta, for underwriting today's educational webcast. You can view the webinar on demand. LabRoots will alert you via email when it's available for replay. That's all for now. Until next time. Bye-bye.